This is showing the base pairing with those hydrogen bonds. So these hydrogen bonds, and let's indicate the partial positive and partial negative. So partial negative on that oxygen to the partial positive on that hydrogen. Partial negative on this nitrogen to the partial positive on that hydrogen. This is attracting this is one DNA strand, and the other side represents the other DNA strand. So there's two DNA strands that are involved in the hydrogen bonds, and that connects two DNA strands. So for cytosine and guanine, there are three hydrogen bonds. So adenine and thymine, this is T and A, those are two hydrogen bonds, and cytosine and guanine, that's C and G, that has three hydrogen bonds.
Even though the shapes of adenine and cytosine are pretty similar, you cannot swap adenine for cytosine. The adenine will never pair with guanine. Same is true about thymine and cytosine. Even though the shapes are pretty similar, it's just the one ring, you cannot swap these two because they're different enough that don't allow for pairing of thymine to guanine or cytosine to adenine. The interesting thing about these bases is that they are puzzle pieces that fit together that drive the creation of the DNA molecule itself. So building this molecule only requires one base at a time. So for instance, this is the side view of a DNA molecule that has, first the top part is just a single strand of DNA that's coiled around. So this is one backbone portion, and then what's sticking out, these horizontal black bars, are the bases. So those bases are A, C, T, or G. Then down below here is the double helix. So that's the already built DNA structure. But it only takes one base at a time to build the DNA because of the fact that the pieces fit together and they drive the formation of this one base at a time. So for instance, if there's a T monomer, a thymine monomer, this will naturally call for an adenine monomer. On the next one up, this is a cytosine monomer, and this will naturally call for a guanine monomer. So here, as we're going along on the side view, you can see that this starts to build the double helix structure as well. So you have the three-dimensional structure. So here's adenine, and this has the same fit for thymine, and also what's shown is that there are two hydrogen bonds that form. Those are those two dots. So here's thymine with adenine. And there are two portions which really fit together well and also are a little bit of a glue together, which are the hydrogen bonds that hold them together. And I'll show you what happens if I try to add a different one of the other bases that is not thymine. So if I try to put guanine, that doesn't work. If I try to put cytosine, that doesn't work because the shapes don't fit. So it's like trying to fit a puzzle piece in where it's not supposed to. If I even try an adenine, that doesn't work either. This only allows for one other base, which is the matching one for adenine is thymine and vice versa. So here's cytosine, and the complementary base that pairs with cytosine is guanine. And you can see that the shape fits, and also those three dots indicate three hydrogen bonds formed between those two bases. So that's what holds this together here, three hydrogen bonds between guanine and cytosine. And I'll try to put something else on there. The shape doesn't fit, and only one shape is allowed, and so this naturally builds. You can see the side view now. This naturally builds on its own, and it is due to the fact that these are complementary base pairs, that each base requires a specific other base to match its shape, and then it's held together by the glue, which is the hydrogen bonding.